Okay, so we're going to be talking about immunology, and our approach is that we're just going to take all of the lectures that we did and we're going to combine it all into one big lecture, okay? This is not exhaustive, so we're not going to cover every single thing, but this is just going to give you an overview of how to, I guess, put it all in one big picture, so then you can go back and you can add little details yourself. So the way I want to do this is I want to talk about viral infection first, and then I want to talk about bacterial infection from beginning to end, from chronologically, okay? So let's just talk about viral infection first. Anytime you have an infection, you're going to be introducing a pathogen into your system. And for viral infections, obviously that pathogen is going to be a virus. And the first thing you're going to do is you're going to activate your innate immunity, right? So your innate immune response goes first. Your innate immunity comes first, okay? And your innate immunity is working for the first 2 weeks. So 2 weeks, okay? So what's happening in that time is you have a virus. This virus is now introduced into your body. And this virus is going to infect a cell. Okay, so now you have an infected cell. All right, so now what happens when your virus infects a cell is your cell is in a stressed state. And the stress state, two things happen. First of all, you're going to decrease the expression of MHC1. Okay, and if you remember, all nucleated cells have MHC1, right? Every single nucleated cell has an MHC1. So what's happening is this virus has been introduced. Let's say that you get a viral infection through your skin. Then you're just going to have a cell floating around in your skin, in your tissue. It's going to be infected. This cell normally has MHC1. It normally has MHC1 on it. But the moment it gets infected by this virus, it's going to stop producing MHC1. Okay, that's the first thing that's going to happen. The next thing that's going to happen is it's going to begin expressing receptors that indicate a stress state, stress receptors. So HLA-E, MIC-A, and MIC-B, okay? So it's going to start producing these stress receptors. And for the sake of simplicity, I'm just going to write HLA-E. Anytime you see that, you know it's going to be any one of these three, okay? And then the infected cell also has on it FAS, okay? And it's always had FAS on it, all right? So your infected cell now has these three things. And in your tissue, you're going to have released NK cells. NK cells. These are natural killer cells, okay? These are the first responders for your innate immune response to viral infection. In fact, they're the only response for your innate immunity to viral infection, okay? So what's going to happen is your NK cell has two important receptors on it. It has KIR and KAR. KAR stands for killing activation receptor, and then KIR stands for killer inhibitor receptor. So just like the name suggests, KAR is going to activate the NK cell, KIR is going to inhibit the NK cell. But KAR and KIR need to bind to things in order to activate, in order to inhibit, okay? So KIR normally, what it's going to do, normally the cell is going to bind to MHC1, okay? Normally it's going to bind to MHC1. But what did we say about MHC1 in an infected cell? We said that you no longer produce MHC1. So what that means is KIR has nothing to bind to. There's nothing over here, which means that there is nothing inhibiting the NK cell. There's nothing inhibiting the natural killer cell. Now at the same time, your KAR, your killer activation receptor, is going to bind to these stress receptors. It's going to bind to HLA-E or MIC-A or MIC-B. So now what you do is you have nothing inhibiting the NK cell, and you have activation of the NK cell. And once you activate the NK cell, it's going to start producing FAS ligand, which, as you could probably guess, is going to bind to FAS. Once you have these two things are bound, let me pick a different color for that. Once you have these two things are bound, and this is not bound, then what's going to happen is you're going to release two enzymes, which will actually do the killing. It's called natural killer cell because it's going to kill the cell. It's going to release perforin and granzymes. Okay, perforin and granzymes. And that's going to result in apoptosis. Apoptosis. And why are you doing apoptosis? Why aren't you lysing the cell? The reason that you're undergoing apoptosis is because a virally infected cell is incorporating the DNA and the viral peptides inside of the cell, okay? So if you lyse the cell, what's going to happen is all of these viral peptides, which are dangerous for the body, are going to be released outside into the tissue. You don't want to do that. You want a very systematic way of killing a cell that does not leak its contents, so it's going to undergo apoptosis, all right? And this is really all there is to the innate immune response for a viral infection. That's really all there is to it. Your NK cells are just going to go and they're going to attack the infected cell. Okay. So then at the same time, what's happening in those first 2 weeks is you begin mounting your adaptive immunity, your adaptive immune response. Adaptive immunity, okay? So in order to mount your adaptive immunity, you're going to have another virus. Because remember, when you get infected, it's not just one virus, it's thousands, millions of viruses that are entering your body at the same time. So you're going to have another virus, and it's going to infect, so to say, a macrophage, okay? And remember how we said MHC class 1 is found on all nucleated cells? MHC class 2 is only found on your professional antigen-presenting cells. So MHC2 on APCs. And what are your APCs? Macrophages, dendritic cells, also follicular dendritic cells, and also B cells. These are your professional antigen-presenting cells. So everything has MHC1, but MHC2 is only found on these different types of cells. So you're going to have MHC2 and MHC1. Okay? So what happens is, let's represent the viral DNA or the viral, essentially the antigen as this pink color. What's going to happen is this virus is going to infect this macrophage, or it's going to try to infect this macrophage. And what the macrophage is going to do is it's going to pretty much chop up the viral peptides that it creates. It's going to chop it up into little pieces, and it's going to present it onto your MHC1 or your MHC2 as well. Okay? So now, at the end of the day, what you want to do is you want to activate a CD8 T cell. Okay? This is going to create a cytotoxic T cell. And this is what's going to do the actual killing. Cytotoxic. Toxic. So it's toxic to cells. Cytotoxic T cells. This is the end goal. The end goal is to create a cytotoxic T cell. But the CD8 T cell cannot recognize the macrophage until it has been activated. So right now it's an inactive, not unactive. It's an inactive macrophage. Okay? So the first thing you have to do is you have to activate the macrophage. In order to activate the macrophage, you need what's called a T helper cell. Okay? So you have a T helper cell. And this T helper cell itself has not been activated, so it's a T helper 0 cell. And you know that TH cells are going to be CD4 T cells, right? Okay. So step 1 of adaptive immunity is you're going to activate a T helper cell. The way that happens is your MHC2 is going to bind to a T cell receptor. This is going to be mediated by the CD4. Okay? That's step 1. Step 2 is you're going to have B7 from the macrophage is going to bind to CD28. Okay? And by the way, B7 is also called CD80 or CD86. All right? An easy way to remember that is 4 times 7 equals 28. Okay? All right. So now you have these two things happen. Your T cell receptor binds to MHC2, your CD28 binds to B7, and the third thing that's going to happen is you're going to release cytokines. Okay? And actually, depending on the identity of the cytokines, it can actually change what the T helper cell is, the TH0. TH0 can become a bunch of different things. It can become actually 5 different things. It can become TH1, TH2, TH17, TFH, or Treg. All right? So what we want to do, and each one of these is mediated by different cytokines. So different cytokines will cause it to differentiate into different things. What we want to do is we want to create TH1. Okay? Let me make that a little bigger. And I need to move this out of the way. Okay? So in order to create a T helper cell, the cytokines that we use is IL-12. Okay? So you use IL-12. IL-12 is released from the macrophage to the TH0. So it goes right here. And that's going to cause the TH0 to become TH1. Now that you have a TH1, the TH1 is going to respond by doing 2 things. First, it's going to release 2 cytokines. One cytokine is going to be IL-12. which is the exact same cytokine that we just released, okay? So this IL-12 is a self-stimulatory cytokine. What that means, okay, let me write that down, self-stimulatory. Okay, what that means is it's going to do clonal expansion, which is another fancy way to say that you're essentially creating more Th1 cells. Okay, and again, these Th1 cells, because they've been activated, remember they're activated by this MHC2. These Th cells are specific for this antigen. It's specific for this virus. Okay, so that's the first cytokine that's released. The second cytokine that's released is interferon gamma, and we'll explain what that does in a second. Okay. So that's the first thing that Th1 does, is it's going to release these two cytokines. The next thing it's going to do is it's going to start expressing CD40 ligand. Okay? Now remember we said that at the end goal, what we want to do is we want to create a cytotoxic T cell. But in order to create a cytotoxic T cell, you need to activate a CD8 T cell. In order to activate a CD8 T cell, you need to activate your macrophage. And right now you have an inactive macrophage. So in order to activate the macrophage, what you need to do is the CD40 ligand needs to bind to this CD40 on the macrophage. And by the way, the CD40 was always there. I just didn't draw it. And now that you have activated, you have this interaction. What's going to happen is your CD40 is going to go, it's going to activate the macrophage. So now it's an active macrophage. And the active macrophage is now going to express more B7. You have more B7. And now that the macrophage is active, your CD8 T cell will now bind to it. So now you have the T cell receptor comes in, binds to the MHC class 1, and then your CD28 is going to come bind to the to the B7, and then you're able to release cytokines. And the specific cytokines that you're going to release are IL-12. Again, you're going to release IL-12. And now you finally created your cytotoxic T cell. Your cytotoxic T cell can now go and it can kill infected cells. Okay, but let's talk about what the cytotoxic T cell does exactly. The first thing it's going to do is it's going to release its own self-stimulatory cytokine, which is IL-2, not IL-12, but IL-2. Okay, and this is again going to result in clonal expansion. Okay, and the next thing it's going to do is it's going to start stimulating Fas ligand. Where did we last see Fas ligand? We last saw Fas ligand up here. Remember, the NK cell starts stimulating Fas ligand when it becomes activated. And we actually said NK cells are a modified type of CD8 T cells. So they're a modified type of CD8 T cells. So CD8 T cells work the same way. When they become activated, they start expressing Fas ligand. Okay? So now you're going to have an infected cell. Your cytotoxic T cell is going to come, it's going to find an infected cell. Okay? So this infected cell is going to have MHC1 on it. And this MHC1 has the antigen on it, right? Right? That should make sense. But actually, remember we said over here that when you're in a stressed state, what happens? Your cell is going to stop the expression of MHC2. So how does this infected cell have an MHC? It's going to stop the expression of MHC1, right? As we said right here, an infected cell is not going to have MHC1 on it. So how did I just draw this infected cell with an MHC1? You remember this interferon gamma? The function of interferon gamma is it's going to go to this infected cell and it's going to tell him, okay, you can start making MHC1 again. It's time for you to start making MHC1 again. So now this infected cell is going to make MHC1, which means that the T cell can bind to it. So you have a T cell receptor over here. This is mediated by, of course, CD8s. The Fas ligand is going to bind with the Fas. And now that you have all of these things come together, you're going to release the same exact enzymes, perforin and granzymes. Okay? And that's going to result in the same thing, apoptosis. All right, that's how you kill infected cells, virally infected cells. That's what you do. So just as a quick review, what you do is you have a macrophage that's been infected with the virus. The macrophage is going to present its antigen onto MHC2 and also MHC1. You're going to then activate a T helper 0 cell. That T helper 0 cell is going to become a Th1 cell. The Th1 cell is going to go back and it's going to activate the macrophage. So it's kind of like you scratch my back, I scratch yours. So the macrophage is going to activate the T helper 0 cell, the Th1 is now going to activate the macrophage. And now that the macrophage is activated, now it can activate the CD8 T cell. So there's a chain of activation. The macrophage activates Th0. Th0 becomes Th1. Th1 activates the macrophage. Macrophage activates CD8. And now CD8 can go and it can kill. Okay? Alright. So now there's just one more thing we need to talk about in viral infection. Okay? And that is, we need to create memory for this. So as long as you have these cytotoxic T cells, you're going to continue to kill this infected cell. And you're going to continue to kill the infection and get rid of the infection. But what happens once you get rid of the infection? What happens once the infection has left your body? You want to create a mechanism by which, if your body gets infected with the same exact virus, you don't need to wait 2 weeks in order to kick off the adaptive immunity. You want to kick off the adaptive immunity right away. Now, in order to do that, what happens is your macrophage is going to stimulate your Th0 cell in a different direction. So notice over here, it uses IL-12 to turn it into Th1. What it's going to do over here is it's going to use different cytokines. It's going to use IL-19 and IL-23. And that's, instead of turning into Th1, it turns it into TFH, T follicular helper cell. And this is going to do two things. The first thing is it's going to release self-stimulatory cytokines. So you see a common theme. The first thing that's going to happen anytime you activate a T cell is you're going to release self-stimulatory cytokines. So that's going to result in clonal expansion. So that these cells can pretty much be their own stem cells. They can create themselves. Okay, so you can multiply them. And the next thing that's going to happen is you're going to present on it CD40 ligand. Okay, so now that you have the CD40 ligand, what's going to happen is you need to create a memory. These T follicular helper cells the whole point of them is to create memory for this exact viral infection. And we know that memory cells come from B cells. So you know that the T follicular helper cell, the TFH, is going to find a B cell. Yep, okay. So we also know that a T follicular helper cell is a type of CD4 cell. And the B cell is a professional antigen presenting cell. So the professional antigen presenting cells have MHC1, because everything has MHC1, but it also has MHC2. Okay? And by the way, this B cell has been activated by the viral infection. So it's actually been infected with this virus, and it's specific for this virus. Okay? So just like over here, in order to put the antigen onto the MHC2 right here, you need this macrophage to be infected by the virus. The same thing happens over here. In order for the B cell to have this antigen on it, it needs to be infected by this exact same virus. Okay? So what's going to happen is this B cell has the antigen on its MHC2. You're going to have the T cell receptor is going to bind to that for the T follicular helper cell. That's going to be mediated by the CD4. And then the CD40 ligand is going to find the CD40 on the B cell. And when you have all of this, you're going to send some cytokines from the T cell to the B cell. And these are the same exact cytokines, IL-19 and IL-23. Okay? And now your B cell can go in one of two directions. It's going to become, you're going to have memory B cells. Memory B cells. And all these are going to do is they're going to go in your bloodstream and they're going to go through your lymph, and they're going to persist in your bloodstream, and they're going to persist through your lymph for the rest of your life. And they have MHC1 and MHC2 for this exact same peptide. Right? So now they can recognize this exact antigen. They can recognize the antigen that we were just talking about. And they're just going to be there forever in your bloodstream. Okay? So that's how you gain memory for this infection. Now the next thing that's going to happen is you're going to create plasma cells. Plasma cells. And the function of plasma cells is to secrete antibodies. To secrete antibodies. Let me make that the same color as the antigen. It's going to secrete antibodies. Okay? And these antibodies are going to be specific for the virus that you created. Now these antibodies can do two things. One thing they can do is they can find the virus and they can coat it. They can just go on top of it and cover it up. And this is called neutralization. This is called neutralization, okay? So it's going to go on top of the virus, it's going to cover it up, and it's going to prevent the virus from pretty much doing anything. It's going to prevent it from infecting, okay? The next thing it's going to do is it's going to go on top of an infected cell. So you have an infected cell over here. And this infected cell, so let me draw it being infected. So you have this infected cell. It's been infected, and we talked about how when you infect a cell, what happens is it's going to start, it's going to incorporate the viral DNA into your DNA. It's going to use your replication machinery, and it's going to make viral peptides. Some of those viral peptides get included onto the membrane. So you have viral peptides that are lining the membrane. Now, what's going to happen is this antibody that's secreted by the plasma cell is going to find these peptides, because these are essentially antigens. It's going to find it. And remember that your antibody has two portions to it. It has the FAB, which is the portion that's specific for the antibody, for the antigen, and you have the FC, which is the constant region, okay? So this antigen-antibody complex is recognized by a cell that we talked about in the very beginning, the NK cell. And this is the last method of killing, okay? What's going to happen is your NK cell has a receptor for the FC portion. It has a receptor for this portion of the antibody. That receptor is called, let me make some more space for this. It's called the FC gamma receptor 3. And you can break this term down. FC is referring to the FC region of the antibody. Gamma is referring to the fact that this is specifically an IgG. Specifically, the type of antibody it is, the isotype is IgG, and the G refers to gamma. So G is for gamma. Actually, let me write that all out. So your different antibodies are IgG, IgM, IgA, IgD, and IgE. Okay? And the specific Greek letters that are going to be associated with it is gamma, mu, alpha, delta, and epsilon, okay? So if you see Fc gamma receptor, you know that it's going to bind to the Fc portion of an IgG, okay? Another name for Fc gamma receptor is CD16. These are interchangeable. Now, when your NK cell recognizes an infected cell that's been coated with this antibody, it's going to release the same exact enzymes, perforin and granzymes, okay? And that's obviously going to result in apoptosis. And this whole process is called antibody-dependent cellular cytotoxicity, my bad. Cellular cytotoxicity, okay? Also ADCC, same thing. So we just talked about the different methods that you use to kill viral infections, okay? So the first method is you're going to use an NK cell to kill an infected cell. That's your innate immunity. That's for the first 2 weeks. The second method is you're going to activate a cytotoxic T cell, a CD8 T cell. And that's going to kill an infected cell. And remember that your CD8 T cells need to activate macrophages in order to activate a CD8 T cell. And in order to activate a macrophage, you need to activate a Th1 cell, okay? And the third method of killing is you're going to activate B cells into plasma cells. Those are going to secrete antibodies. Those antibodies are going to coat infected cells, and that's going to recruit NK cells, which are going to kill via antibody-dependent cellular cytotoxicity. So these are the 3 methods of killing in a viral infection. And that's it.